sequences means they are been manually curated. People like us have or like you have sat together and have clearly looked into the sequence that everything is correct. It is a novel gene, it is a novel protein, and then have tried to annotate it and then have put it in the reviewed database, reviewed part of it. If it is unreviewed, the shown by blue file here, which is part of TreeMBL, which is translated EMBL. So whatever sequences are, DNA sequences has been submitted so far are translated in different frames and has been submitted in unreviewed section, which has not been manually curated yet. So probably in future, some of these sequences will move to the reviewed section. So this is one other resource where you can get protein sequences downloaded. I can't see the slide anymore. So we talked about GenBank database from NCBI, okay, NIH, and other database what we talked about was UniProt. So when you're working with protein sequence, you go to UniProt database. When you're working with DNA sequence, you go to the GenBank database. I hope this point is clear to all of you. Dr. Vivek, are you there?
He is not there, ma'am. Maybe some network issue.
Okay. No problem.
He joined. Yeah, he joined.
He joined? Okay.
Yes.
I have just one more slide, one or two slides.
Okay.
Manjari, can you see the slides?
Not yet, Vivek. Yeah, now it is visible.
Okay.
Now, can you go to the next slide, Vivek? I'm almost done. So students, from today's part 2, if you can take this message back home, that whenever you get time to sit in front of computer, go to these two databases, GenBank and UniProt, and type a gene of your interest and see what sequence information you are getting. Try to download the FASTA sequence of it, and once you get the FASTA sequence, try to learn how to download the genome sequence. Now, I talked a lot about human plants, but are there specific databases for specific model organism? Yes, there are. For example, for Drosophila, you have Flybase. So those who are interested in working with Drosophila should have a look on Flybase database. Then for mouse, MGI is another popular database to get the genome sequence, to get the gene sequence there. Then for zebrafish, it is ZFIN, the zebrafish model organism database. For Saccharomyces cerevisiae, it is SGD. And then for C. elegans, it is Wormbase. So depending upon your interest and then which model organism you choose to study a behaviors, to study a particular pathway, you go to that database, retrieve the sequence and start working on it, which you will be learning from Dr. Shashi's talk, that how you can manipulate the genome. So before manipulating the genome, you also should know the sequence, and these are the places from where you can download the sequences. So my last slide is, these are probably the recent trends in biological research where you can think of designing your project or think of reading more papers. So I think because of the availability of lots and lots of data and because of different databases, one trend which has been from last many years, it has been growing and will also be in demand in future, is coding in biology. So if you're interested in learning coding in any programming language like C, Perl, Python, so have practice of or start taking those classes. These programming languages will help you to understand data pretty well. You can integrate data from different places. You can prove any hypothesis by working on the data sets. So coding in biology is one very important trend, and it will be again in demand in future as well. Then there is next generation sequencing and different platforms available. Now you have lots of information in just few days, or maybe by very minimum amount of material being used. So because of more and more of NGS data available, and because of lots of high throughput experiments nowadays available, you will again need coding in biology. Dr. Shashi will talk about genome manipulation, that I will not talk about that much. Then you have more of genograph experiments now, as Dr. Vivek talked about different model organisms. So when you set one hypothesis, you also need model organism to test it, and you'd go for genograph experiments there. And finally, all of us always not just want to understand the principle or the basic biology, but we also want to bring biology or to bring research to that extent that it can be useful for patient. So finally, the recent trends are also moving towards personalized medicine, using genomics data to cure a particular disease. So we want to bring bench to bed kind of research. So with that, I would like to end my part, and Dr. Shashi will take over from here. Thank you, Vivek, for sharing the slide.
Okay, so should I stop sharing?
Yeah, you can stop sharing.
Yeah.
Thank you.
Yeah, thank you so much, ma'am.
Thank you.
Hello, I am Dr. Shashi.
Welcome, sir.
Well, thank you. So the share option is not coming for me also.
Sir, I think. Screen is visible, sir. But screen is visible, sir. We can see it.
You can see my screen?
Yeah. Yes, sir.
Okay, good. Okay, so you can see my screen, right?
Yes, sir.
Good. So it has been a lot of data talks in previous talks by Vivek and Manjari. So you can take an idea from the previous talks that how much work has been done to collect data from for our DNAs, right? So because all the information what we think is coded in the genome or the DNA, what we have, right? And DNA is present in each and every cell of the body. So now with a lot of different techniques coming in the last century and a lot of computational work, like computer science coming in this field, we have a lot of information, and we know about almost every gene present in humans and the model organisms which Vivek talked about, right? So like how are we going to make use of this information? So you are lucky that you are born in this time of the history, because several changes are taking place, and now we'll talk about a very recent technique which has come and which is going to revolutionize the way we are going to see the living systems. Okay, so before talking about the science, let me show you some movie titles. So you must have seen some of these movies, and if you see many of them, you can correlate, at least you can have some idea that these movies are all themed on a genetic disease, right? And very often we wonder how why these movies are getting made, and how common are these genetic mutations, right? And how it affects individual. So I think these diseases are the main reason why we are studying, why we are going deep and deep into our DNA and trying to know what is wrong with a person's DNA, like him, what happened, what is wrong with his DNA, that he got a big foot like this, or why somebody got slow mental growth. So all this information is there in the DNA, but and we know which genes are doing it. But can we correct these people? Can we make them disease free? So why this is important is this is not a small number of people. If you consider the whole human population, 10% of human population, or 0.8 billion people, are suffering from one or other form of a rare genetic disease. Okay, so several of them are there, and Dr. Ashwin will talk more about these diseases. Okay, So what happens? So here is an example of one of such diseases. This is a sickle cell anemia disease seen in some people. And what happens in their DNA is there is a mutation in one of the mutation means change of base. So normal people will have certain DNA sequence at a particular position in a particular gene, and in the diseased person that sequence has changed, and very often these changes are just one letter long. So this change of one letter makes a person diseased. Okay. And this is something which we want to correct. Right. So, like if this change is reversed, the person can get all right. Right. So science is aiming to do that, and you can see in the left side just now, Manjari was talking about how genes are arranged on the chromosomes. Right. So there are different diseases arranged which comes from different chromosomes, and there are a number of them. So although we say like most of the people have good health and we think we are healthy, but if you look into our genomes, we still carry some copies of defective genes, but they are in right combination that we do not produce disease. But maybe our future generations can have these mutations, and that may be manifested in a disease, right? So that possibility is also there. So the whole scientific exercise which we do, the goal is to somehow correct these changes, correct these mutations. So this is a slide where I'm showing what if it was possible to remove the faulty base and replace it with the correct one, like this, like the cartoon it is shown here. Why can't we do this? So scientists have been thinking this for last 50, 60 years. And why I said that we are lucky, that because only 10 years ago, a technique came where we could do this. So precisely, we could remove a faulty base and replace it with the correct base. So the technique was known as Crispr. I am sure most of you or many of you would have heard about it, because it was discovered in recently, like 10 years back, and last year it got the Nobel Prize. Okay. So it made such a tremendous impact on the life sciences that this discovery made to Nobel Prize within 6 or 7 years of its discovery. And why it made such an impact? Because of its like potential, right? So with this, we can correct these. We have a hope that someday we will be able to correct genes in these people. Okay, so I will give you a brief idea about what this technique is, and you can go ahead and contact me or go and read more about it. So the CRISPR stands for clustered regularly interspersed repeats. Each of these words have a scientific meaning. I'm not going into it, but just giving you a first exposure to what this technique is. So this technique is something like it behaves like scissors, and you know DNA has 2 strands, and it cuts both the strands, and it can cut where we want exactly. Okay, so it has 2 components: a guide RNA, which is a RNA, and a protein called Cas9. The guide RNA acts like a tether. So you must have seen people walking their dogs with a tether, right? So it acts like a tether. So wherever you want, you can take this dog with this tether. Similarly, with this guide RNA, you can take Cas9 anywhere on the DNA you want. And because there is a sequence specificity, so if you want to cut a particular gene, you can make a guide RNA for that particular gene, and it will go and cut at that particular place. Okay, and the Cas9, which is the protein, that's the actual scissor. Okay, so that will cut the DNA. So with these two simple components, now we have the capability to to edit our DNA at any location, and that gives us a great hope. So here I am showing the sgRNA is like the tether and the Cas9 is like the dog, which actually does the job. And after these cuts are done, we can have a repair template, like the DNA which we want to correct, the corrected sequence here, and this goes in the DNA and integrates and corrects the sequence. Okay, so it's like a cut, copy, and paste mechanism. So you cut whatever is not desired, copy whatever is desired, and paste it. And now we have the ability to do it very quickly and anywhere in the genome. Okay, at least in the experimental setup. And why is this so popular? Because although there were such methods earlier, but it was not possible to do it in every lab. Now with this method, we can do it in every lab at a very high speed. Like you can do an experiment within 3 days. You can see the effect within 3 days of your experiment, and it is very accurate. You can cut precisely where you want, and it is very simple. You just have to put these cells in the put these things in the cells. That's called a transfection, a small step in the practical life sciences. And with this, you can edit your genomes. Okay. However, although we have got the tool, but the major hurdle now we have is, should we edit our genome? Like, should we play God? Like, what makes us human is our genes, right? So should we start editing it? And that's a very vital question, which like all the policymakers and all the big leaders are debating upon. Like, should we use this tool for editing? So, and if we want to use it for editing, where should we draw the line for gene editing? Like this tool can be used for curing diseases, or it can also be used for aesthetic reasons. Like if somebody wants to be taller, he can edit his tallness gene. If he wants big muscles, he can do that with this tool. So should we allow that? So it's a debate going on, and how far should we allow that? So that's limiting the use of CRISPR presently in humans. But I will just show you in the next slide where it has been used and how it is still useful. And also, can parents be allowed to edit their babies which are still to come? So if a parent wants that my son or daughter should be very tall, and that is completely possible by CRISPR. Should we allow it? We don't know. So that's the discussion we should have, and I'm sure your generation will see the effects of this technique. Okay, so let's see what has been done. So as I said, the CRISPR has not been used for editing in normal humans till now, but there are some critically ill patients which have very low chances of survival, right? Like there are cancer patients. You know, you must have heard that cancer is such a enormous disease, and many people get it, and several people die from it, right? So how CRISPR has helped in the survival of these patients? So in the cancer patients, there are some like in all people, like in us, in you, in everybody, there are T cells which fight cancers. Okay, so if the cells are trying to become cancerous, your T cells will kill them normally. So that's why we don't get cancers. But in people who have got cancers, these T cells have been weakened. They are not able to do their functions very efficiently. So what scientists have done with these T cells, they take out these cells, they edit it with CRISPR so that the genes which we are protecting, which are protecting us from cancer, those are put back or made more active. And then those cells are grown outside the body, outside the human body. Have I lost you? Okay.
No. Yeah.
No, right?
No, sir, you are there.
So these cells are grown in large numbers outside the body, and then they are infused back in the patient. Now these T cells, what they will go and do? They are again active. Okay, so they will start killing the cancerous cells. Okay, so this is the type of application which has come up and which is actually in the clinics, and people are getting benefited from it. And although it is costly in the present day, and it's unfortunate that many people cannot afford it, but we have got the technology. So I'm sure in coming days we will see more applications for it. And I will give you another example. So at least some of you might have heard that uh, that somebody has loss of kidney function, somebody's heart failed and he died. And somebody is in critical condition waiting for for an organ, like somebody is waiting for kidneys, heart, right? So and there is severe shortage of such organs because, of course, because of the obvious reasons, right? So one solution is use the organs from the animals. And how this is possible? This is again, so normally if you use an organ from an animal, what will happen is your body will reject it because animals we have a very different immune system. Okay, so our body will recognize something from outside as foreign, and they will not take it. So if you take kidney from a pig and put it in a human which has a defective kidney, it will not start working. It will be simply rejected by our body, saying that this is not my kidney. I should not accept it. This can be something dangerous, right? So this is being addressed currently with the CRISPR technique. So now what is being done is, in an embryo of pigs are modified genetically, or their genes are edited so that they do not look like pig organs anymore. The markers or the things which make it more pig-like, they are made to make look like human. Okay, so the genes are changed in such pigs such that it looks like more human. Now, if such kidneys are implanted or transplanted in humans, that kidney can be functional and are functional actually. So this is one a life-saving procedure which is possible, and it has not still come in clinic. But recently we saw that the first heart transplant was made, right, on January 7th, 2022, where a pig heart was transplanted into a human. The patient was terminally ill. His heart has failed, and there was no hope for him to survive. So they allowed this experiment on him. That let's see what happens. And fortunately, this has worked, and we have first human being who has survived with a big heart. So these are very great advancements which we are looking at, and we have like these are all first times in the histories, and hopefully they will make big marks in the years to come.
Sir, is it good, sir, to have such type of hearts?
So what do you think? If somebody is dying, should we allow him to lose his life just because of one heart failure?
Sir, we can't know, sir.
Yeah. So as I said, these things are still not approved in clinics. People are debating, like, should we have this? Should we not have this? What can be its implication? So it's still in debate. But what I'm trying to present here is these techniques are here, and we should start thinking about it. That should it be used to help patients, or we should just keep it confined to laboratories? Okay, at least in the disease condition. Yeah, tell me.
Can we retransplant? Can we retransplant the organ if our body rejects transplanted organs?
can we retransplant to some other human? Sorry, I did not get your question.
Sir, can we retransplant the organ if our body rejects a transplanted organ?
Okay, so yeah, it has never been done, but I think if the patient is otherwise healthy, it is possible. So what happens mostly is if an organ rejection happens, the patient becomes very sick, and his other systems also starts failing, right? But if the patient is still remains healthy, I think we should have he has another chance of transplantation. And in this context also, like if you think of transplanting a human transplant, doing a human transplant, say, so if you have transplanted somebody with a heart and it has not worked, it's very difficult for that person to get another heart from another human, right? Just because of the heart. Yes, sir.
Yes, Kishore, you want to ask some question?
Yes, sir.
Yeah, please.
Yes, madam. Why a pig? Is it because there is already some similarities? Or can theoretically any animal which is similar in size perform such
No.
transplant?
So, yeah, so pigs are more suitable for the anatomy of the
Pandey,
pig
you mute
organs.
yourself.
The anatomy of the pig organs are most similar to
Sir,
Other students, please mute yourself. Shivam Pandey, please mute yourself. Yeah.
okay.
Yes, JNV G2, yes.
Yeah.
Please answer.
Can we have a library of vital organs formed from our stem cells?
Pardon? Can you repeat?
Can we have a library of vital organs formed from our stem cells?
Can we have a library of organs from other
Can we have a library of vital organs formed from our stem cells?
organs from stem cells? So till now, has not achieved. To like take the cells from the patient and grow it into full organ in a short span of time. Because when organ failures happens, we don't have a lot of time to grow that organ, right? So that's an option, but hopefully someday we will have that option too.
Yeah, 1 minute. You guys are raising your hands, but before asking the question, make sure there is no noise, okay? Because it's not clear. And ask your question properly. And after asking the question, please mute yourself. Now you can ask your question, Kishore.
Madam, sir, why was the pig chosen as the recipient for the heart transplant? Is it because there is already some similarities, or can theoretically any organ's heart be transferred if there is structural similarities?
So, so the work on the transplantation has just begun, and why they chose pig? There are many reasons. The first and most important reason is the organs of pigs are anatomically and the size wise very similar to humans. Okay, so if you consider the anatomy of pig heart, it's very similar to a human heart. So sizes and anatomy is very similar. So that's one reason, and the second reason is like pigs have lot of offsprings. So once you breed a pig, you can get up to 7 or 8 offsprings. Okay, so that way, also if you have a genetically edited pig once, you can propagate it very fast, and their gestation time is just 3 to 4 months, right? So in 3 to 4 months, you can make from 1 pig to 10 pig. But if you take a larger organism, yeah. So those are some of the reasons. Yeah. Any other question?
Sir, I have a question. So if now the person who has got the transplant of heart of the pig, sir, will he be suffering of any side effects of transferring a heart, sir?
Madam, will he be?
Sir, now he has got transplanted heart of pig, sir, so that by differentiates between human heart and pig heart, sir, will he get any side effects of it, sir?
Side effects? Yes. So that we currently don't know. So as I said, only the first transplant has been done. So only after such transplants become common, we will learn that what side effects are there. Definitely, there will be some side effects, but it's better than death, right? It's like people will take it rather than dying away, right? So yeah, so that's the second thing. And once we are doing it more regularly, we will have side effects and we will have remedies for it also. That's all science about, right? We keep getting problems and we keep solving it. Okay.
Yes, sir. Thank you, sir.
Any other questions?
Sir. Sir, that heart was taken from a dead pig or an alive pig, sir?
So after taking the heart?
No, sir, that heart was taken from an alive pig or from a dead pig, sir?
Sir, you are mute, sir. Shashikiran, sir, you are mute, sir.
Am I unmute now?
Yes, sir. Yes, sir.
Yeah, so the heart was taken from a live pig. That's what I was saying. Okay. So, yeah, because the heart needs to be functional, right?
Excuse me, sir.
Yeah.
Is there necessary to match the blood sample of pig and human body?
The blood sample. I think no, because they will not match.
Sir, without blood matching, how we can transplant, sir?
Okay, so for that you should understand why blood matching is done. So blood matching is done to match the antigens, right? So I was talking about immune cells, right? So when we talk about A-type blood, B-type blood, what happens is A-type blood has certain antigens which will be considered foreign by the B-type blood person. Okay, so that's happening in humans. So when we match blood among humans, that's the typing. But pigs might have, I'm not aware of that, pigs might have a different system of blood typing. They may not have A, B, and O types. So, and
which antigen present in pigs, sir? Antigen.
I'm not aware of that. I'm not aware of that.
Okay, students, actually, we can understand that you have many questions, but sir, next our ma'am is waiting for, so it's.
Sure. You can have post your questions to me later.
Yeah. Yes. Later on, they can post.
So I will stop here. I will just show you that last slide. So the applications in humans that are going on. But we have some degree of freedom to work on animals. So accordingly, we have several animals being made by genome editing. And these animals are engineered so that they are more helpful to humans. Like this goat has been made so that it has less fat in its meat. This calf is such engineered that it will produce only male offsprings, no female offspring. So it will produce only bulls because bulls are good for meat in Western countries, right? So this calf has been made. This dog has been made to have more muscles. Okay, so there is the muscle growth the gene which stops or retards muscle growth, that has been mutated here. So similarly, other animals have also been mutated. Okay, so as I hope I have drawn your attention to this technique. You are free to write to me anytime of.
Sure, sir. The students, you can send your queries to JNV Rangareddy and we will forward it to Sir Shashikiran, sir. Okay, beta. Now, yes, sir.
Can I stop? I am with my presentation, ma'am. Hello.
Please mute yourself, others. Please mute yourself. Shashikiran, sir, if it's possible, can you please share your mail ID to us so that if there are any queries, they can directly ask you, sir.
Sure, ma'am. Sure.
Okay, sir.
I'll text you then.
Okay, sir.
So thanks, children. Thanks for listening this. And so what should I do?
Yes, sir. Thank you so much, sir. Thank you, Manjari ma'am. And Vivek, sir, I think sir left. Thank you so much for this interactive session. Thanks a lot. So next in line, we have our Dr. Sarita Wig, ma'am. And students,